Okay. All right. So for part A, it was asking what is the what the banding pattern will look like on the gel, and I don't know if you can see that, but we do have that one written up here on the board for 1A, and so that's what it would look like for that. And
Is the map not on the screen? Oh, there we go.
yeah. All right. And so for part B, it's asking, suppose you use E. coli DNA polymerase 1 during the sequencing reaction instead of the Taq polymerase. And so we came up with the answer that the E. coli DNA polymerase 1 will denature during the heating process, so the DNA strands will not be synthesized when cooled. And then for part C, suppose I accidentally add E. coli DNA helicase to the reaction in addition to the normal DNA sequencing reaction components. And the DNA helicase shouldn't be a problem as it will probably denature during the heating and it shouldn't affect the rest of the process very much. And then for D, suppose I add DDATP to one of the 4 tubes. However, I forget to add the DDGTP, the DDTTP, and DDCTP to the other tubes. Assume the other components used for sequencing are normal, and only the test tube with the DDATP will have incomplete, is that right? Incomplete DNA sequences. The other test tubes will have fully synthesized DNA strands because they don't have any DT NTPs to terminate replication. So the first tube will show whenever there is a DD ATP in the sequence, but we won't know the other sequences. And then for question 2, with the fill in the blanks, during a typical PCR cycle, the Taq polymerase synthesis of adding DD NTPs step is done at 72 degrees, while the primer annealing or binding step is done at 55 degrees. And part B, AMP subscript R is a common selectable marker that is found in many plasmid vectors, while restriction into endonucleases recognize and cleave DNA molecules at palindromic sites. And then part C, during agarose gel electrophoresis, DNA is placed in an electrical field. DNA migrates towards the positive electrode during electrophoresis. And part D, DDATP lacks the 2' OH group and the 3' OH groups. E, transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. And then F, during blue-white screening, IPTG is commonly used to induce expression of the LAC operon, while XGAL is cleaved by the beta-galactosidase to turn E. coli cells blue. And then for question 3, you are interested in using PCR to amplify the region of a double-stranded DNA fragment from E. coli. The sequence to be copied is located between the sequences in bold below. And so for part A, we also have those written on the board. So it is asking, write a single-stranded DNA sequence that you would use for the 2 primers used in the PCR experiment. Note that each primer should anneal to one of the sequences in bold. And so we have those written up here for part A. And then for part B, it's a mathematical equation can be used to determine the annealing temperature that should be used for PCR. This question is, or this equation is the annealing temperature in degrees Celsius 81.5 plus 0.41 times the percentage of the G's and C's minus 675 divided by the total number of nucleotides. And then to get the annealing temperatures that we would predict for each primer from part A, for the first one, we found that there is 56.25% G's and C's, and that there is a total of 16 nucleotides. So we put those in the equation and we got 62.4 degrees Celsius. And then for the second strand, we found that it was 50% G's and C's. And then again, with a total number of 16. And so we put those in and we got 59.8 degrees Celsius. And then for part C, each PCR cycle contains a DNA synthesis step. What are the sequences of the two newly synthesized strands if DDCTP is accidentally included in the PCR experiment? Assume that DATP, DTTP, DGTP, and all other appropriate PCR components are found in the reaction mixtures. So for the first one, we got 5' with the primer, and then GGGATTAGC 3'. And then for the second one, we got 3' CT. Well, if you read it from 5' to 3', you'd have the primer, and then TTTAATC, and then 3'. And then for part D, when we learned about DNA replication in E. coli, we learned about DNA A and DAM, and why are these two proteins not included in a PCR experiment? So DNA A is a protein that binds to double-stranded DNA, bends it, and then allows the polymerase complex to separate to the two DNA strands. Because PCR is done at a high temperature, DNA strands dissociate freely, so that primers can just find the right annealing site and bind without proteins to assist them. And then part 2 of that, the DNA adenine methyltransferase methylates specific regions of DNA strands to prepare them for replication. Without adenine methylation, replication will not take place in normal situations. In PCR, true replication isn't taking place because the double DNA strand is being split by temperature and not the replosome.
Sweet. Okay, and then for question 4, concerns agarose gel, electrophoresis, and restriction mapping. Suppose you set up 6 restriction enzyme digestion reactions. Each reaction contains the same linear DNA molecule, but in each case, the DNA molecule is digested with different combinations of the restriction enzyme. The 3 enzymes are the BAM-HI, ECO-RI, and HIND-III. And then it gives a table of the different things. So the first thing was to draw draw the results of agarose gel electrophoresis. And I don't know if you can see it. Let me make sure you can see it. It's right. That's what we have right here. So this is the marker, and this is the different. This is 100 to 1,000, and these are the tubes 1, 2, and 3. Each of these have just one of the restriction enzymes, and then 4, 5, and 6 have a combination of 2 of the enzymes. And so we have it on the the Word document as well that we'll turn in. And then draw a linear restriction map that indicates the location of the restriction enzymes, of the restriction enzyme sites along the hypothetical DNA molecule. Make sure you can see that over here. So over here it shows so over here it shows there's 50. It's a 50 base pair segment, and then there's the BAMHI restriction enzyme site. Then 150 base pair segment, and then EcoRI restriction enzyme site. 100 base pairs, the HIND3, or I don't know if those are threes or I's, and then the next is 100 base pair unit, and then again the BAMHI, and the last section is 500 base pairs long. And then see what are the predicted fragment lengths, and if this linear DNA molecule is cleaved with all three molecules simultaneously. So that would be just the different segments, because this shows all of the different sites. So if we use all of the enzymes, you would have a 50, 150, 100, two 100s, and then a 500 base pair long segments. And then for 5, we are studying our favorite gene again, and we identified it. We identified it in yeast, and so it says detail the steps that you would use to generate cDNA from the mature mRNA, clone a cDNA version of the YFG into a plasmid vector, and use the blue-white screening to identify plasmids that contain the YFG. So first, we would need to purify the mature RNA that contains the YFG. Then we would need to mix the mRNA with a primer, like a poly-DT primer that corresponds with the poly-A tail at the end of mature DNA or mature mRNA. Then we need to add reverse transcriptase along with a bunch of the dNTPs so that it could synthesize it backwards, starting from the primer that has lots of T's. And then the reverse transcriptase that makes the complementary strand of the DNA that matches the mature mRNA. Then we use another enzyme called RNase H to digest the majority of the RNA. And then, so most of the mature RNA that we has now been RNase, and so there's we don't have an RNA DNA hybrid like we had before. We just have the DNA that was transcribed by reverse transcriptase and the RNA primers at the priming ends. And then finally, we add DNA polymerase 1 to synthesize the rest of the complementary strand. So now we have the 2 of DNA. Then we take the newly synthesized DNA and insert it into a host cell through transformation. And once transformed, or once transformation is complete, we allow the host cell to multiply, and so it multiplies the plasmids. And then we put the cells on a medium that have IPTG and XGAL to see, so if the new gene was taken in and inserted where the LAC-Z gene is, then no beta-galactosidase would be made, so no XGAL would be metabolized, and the cell wouldn't turn blue. But if it didn't take it in and disrupt the LAC-Z gene, then the LAC-Z gene would continue to make beta-galactosidase because of the IPTG. And when beta-galactosidase is present with XGAL, it metabolizes XGAL and turns it blue. So that's how we could tell whether it was taken in or not. And then for 6, the CRISPR-Cas9 is a relatively new technique in molecular biology research, and then you want us to research the technique and write a paragraph about it. So CRISPR-Cas9 is pretty cool, but it's just a way that geneticists edit DNA. Or they disable a gene, or they can insert a whole healthy gene and take out a bad one. But basically, it's the natural way that bacteria protect themselves from invading viruses. So the virus injects its DNA in the bacteria, makes two RNA strands. One of them matches part of the invading DNA, and the other one is just important in forming the Cas9 complex. So the two RNA strands, they combine along with a protein, and all three of those things make the Cas9 complex. And then this complex goes and finds the site that the RNA matches the viral DNA, and once it matches it, it kind of inserts the RNA into the DNA so that it's bound through its nitrogenous bases, and then the protein then cuts the viral DNA, and so it renders it disabled, I guess, and it won't harm the bacteria anymore. But recently, the Cas9 complex has been engineered to cut any sequence of DNA by modifying the guide RNA within the complex. And what's cool is it doesn't just work in like test tubes, but they can actually put Cas9, the CRISPR-Cas9, into a living nucleus, and it'll look for the DNA that they've that the guide RNA corresponds to, and it'll go cut any kind of DNA. And it's kind of cool because it can cut out a section, and then they can put in a healthy copy of the gene that will go find that cut out section and kind of mend it back together. But yeah, basically that's all I found on the CRISPR-Cas9. It's a pretty cool thing. So that's our video for lesson 13.